Microbiology for GNM Unit 4 Immunity, Types of Immunity, Innate and Acquired Immunity. Immunity is the body's ability to defend itself against harmful pathogens, bacteria, viruses, fungi, toxins, using defense mechanisms. Types of immunity: innate immunity, natural non-specific, present from birth, provides immediate non-specific defense through barriers and immune cells. Acquired immunity, adaptive specific, developed after pathogen exposure, provides long-term specific protection. Innate immunity components: physical barriers, skin, mucous membranes, cilia, chemical barriers, lysozyme, tears, saliva, gastric acid, interference, complement system. Cellular components: neutrophils, macrophages, phagocytosis, NK cells destroy virus-infected cancer cells. Physiological barriers: fever inhibits pathogens, inflammation, immune response activation. Acquired immunity types: active immunity, develops after antigen exposure, long-term protection. Natural, occurs after infection, e.g. measles recovery. Artificial, developed through vaccines, e.g. polio vaccine. Advantages: long-lasting memory cell formation. Disadvantages: takes time to develop, mild side effects. Passive immunity, ready-made antibodies, immediate but short-term protection. Natural, maternal antibody transfer, IgG via placenta, IgA via breast milk. Artificial, injection of pre-formed antibodies, e.g. tetanus antitoxin, rabies immunoglobulin. Advantages: immediate protection, life-saving in emergencies. Disadvantages: short-term, no memory cell formation, possible allergic reactions. Conclusion: active immunity offers long-term defense but takes time, whereas passive immunity provides immediate but short-lived protection. Both are essential in nursing for infection control, vaccination and emergency treatments. Immunization Schedule and Immunoprophylaxis, Vaccines and Sera. Immunization Schedule. Immunization is the process of providing immunity to an individual against infectious diseases through vaccines. The schedule helps protect infants, children and adults from preventable diseases. Immunoprophylaxis. Immunoprophylaxis refers to the prevention of infectious diseases through the administration of vaccines or immune sera. It involves using vaccines to induce active immunity or using immune sera to provide passive immunity to protect individuals from infections. Active immunity, vaccines, stimulates the immune system to produce its own antibodies after exposure to a weakened or killed pathogen or its components. Passive immunity, immune sera, provides immediate protection by transferring pre-formed antibodies to an individual, offering short-term defense against specific infections. Vaccines. Definition: a vaccine contains weakened or killed antigens that stimulate the body's immune system to produce antibodies without causing disease. Examples: BCG protects against tuberculosis, OPV protects against polio, DTP protects against diphtheria, tetanus and pertussis. Hepatitis B protects against hepatitis B, measles, mumps, rubella, MMR, protects against measles, mumps and rubella. Hepatitis A protects against hepatitis A. Sera. Definition: sera contain antibodies or antitoxins that provide immediate protection against specific infections, offering passive immunity. Examples: tetanus immunoglobulin, TIG, given to individuals who have suffered from a potential tetanus-prone wound. Rabies immunoglobulin, or RIG, given after a bite from a rabid animal. Anti-venom, given after venomous snake bites. Hepatitis B immunoglobulin, given to newborns of mothers with hepatitis B. Conclusion: vaccines stimulate the body's immune system to build active immunity for long-term protection. Sera provide passive immunity and are used for immediate protection. Following the immunization schedule is vital to ensure protection from infectious diseases. Hypersensitivity and autoimmunity. Hypersensitivity is an exaggerated or inappropriate immune response to an antigen, leading to tissue damage or dysfunction. It occurs due to the immune system overreacting to harmless substances, causing pathological effects. Types of hypersensitivity, Gell and Coombs classification. Hypersensitivity reactions are classified into 4 types based on immune mechanisms and onset time. Type 1, immediate hypersensitivity, anaphylactic reaction. Mediators, IgE antibodies, mast cells, basophils, histamine, leukotrienes, prostaglandins. Onset, rapid, within minutes of antigen exposure. Examples, allergic rhinitis, hay fever, asthma food allergies, e.g. peanuts, shellfish. Anaphylaxis, severe allergic reaction. Mechanism, sensitization. Phase, first exposure to allergens stimulates B cells to produce IgE antibodies. IgE binds to mast cells and basophils, sensitizing them. Effector phase, re-exposure. Re-exposure causes allergen cross-linking of IgE on mast cells. This leads to mast cell degranulation, releasing histamine and other mediators, causing vasodilation, hypotension, increased permeability, swelling, edema, bronchoconstriction, wheezing and difficulty breathing. Treatment: epinephrine for severe anaphylaxis. Type 2, cytotoxic hypersensitivity, mediators, EGG, IgM antibodies, complement system, natural killer NK cells. Onset, hours to days. Examples, hemolytic disease of the newborn, Rh incompatibility, blood transfusion reactions, autoimmune hemolytic anemia, myasthenia gravis mechanism. IgG or IgM antibodies bind to antigens on target cell surfaces. Complement activation leads to opsonization, marking cells for phagocytosis, membrane attack complex, MAC formation, cell lysis, NK cell-mediated destruction. Example, mismatched blood transfusion causes donor RBC destruction, leading to hemolysis. Type 3, immune complex-mediated hypersensitivity, mediators, immune complexes, antigen-antibody, complement proteins, neutrophils. Onset, hours to days. Examples, serum sickness, rheumatoid arthritis, systemic lupus erythematosus, SLE, post-streptococcal glomerulonephritis. Mechanism, soluble antigens combine with antibodies forming immune complexes. Complexes deposit in tissues, e.g. kidneys, joints, blood vessels. Complement activation leads to neutrophil recruitment, release of inflammatory enzymes causing tissue damage, increased permeability, localized swelling and damage. Example, in post-streptococcal glomerulonephritis, immune complexes deposit in kidneys, causing inflammation. Type 4, delayed type hypersensitivity, cell-mediated hypersensitivity. Mediators, T lymphocytes, CD4 plus T cells, CD8 plus T cells, macrophages, cytokines. Onset, 24 to 72 hours, delayed response. Examples, tuberculosis, TB skin test, contact dermatitis, e.g. poison ivy, latex allergy. Type 1 diabetes mellitus, organ transplant rejection. Mechanism, sensitization phase. APCs, e.g. dendritic cells, present antigen to T cells, memory T cell formation. Effector phase, re-exposure. Memory T cells release cytokines, e.g. interferon gamma, tumor necrosis factor alpha. Macrophages get activated, release of inflammatory mediators, tissue destruction. Example, contact dermatitis occurs when T cells trigger inflammation at the skin site in response to allergens like poison ivy. Management of hypersensitivity reactions, avoidance of allergens, e.g. food, drugs, environmental triggers. Antihistamines for mild allergic reactions, corticosteroids to reduce inflammation, epinephrine injection for severe anaphylaxis, immunosuppressive therapy for autoimmune hypersensitivity, desensitization, allergy shots to modify immune response. Conclusion: hypersensitivity reactions result from an exaggerated immune response and can range from mild allergies to life-threatening conditions like anaphylaxis. Early diagnosis and proper management are essential to prevent complications. Autoimmunity. The immune system attacks the body's own cells, causing inflammation and tissue damage. Causes: genetics, environmental triggers, hormonal influences, and immune dysregulation. Types of autoimmune diseases. Organ-specific, type 1. Diabetes, Graves' disease, Hashimoto's thyroiditis, pernicious anemia, myasthenia gravis, systemic, SLE, rheumatoid arthritis, multiple sclerosis, scleroderma, Sjogren's syndrome. Mechanism. Loss of self-tolerance leads to autoantibody production, T-cell activation, chronic inflammation, and tissue damage. Diagnosis and management. Blood tests, biopsies, imaging, treated with immunosuppressants, symptomatic relief, lifestyle changes, and plasmapheresis if severe. Prevention and care. Early diagnosis, timely treatment, regular monitoring, and patient education. Conclusion: hypersensitivity and autoimmune diseases arise from immune dysfunction and require early detection and management. Principles and uses of serological tests. Serological tests are laboratory diagnostic methods used to detect antibodies or antigens in a patient's serum. These tests help identify infections, immune disorders, and blood compatibility by analyzing the body's immune response to pathogens. Serological tests detect antibodies or antigens in a patient's serum to diagnose infections, immune disorders, and blood compatibility. Types of serological tests. Agglutination tests. Principle: insoluble antigen and specific antibody interaction leads to visible clumping or agglutination. Those clumps are called agglutinins. Uses: slide agglutination, blood grouping, bacterial identification, tube agglutination, Widal test, typhoid fever diagnosis. Latex agglutination detects C-reactive protein, CRP, rheumatoid factor, RF. Precipitation tests. Principle: soluble antigen and specific antibody forms complex of an insoluble precipitate. The process is called precipitation. Uses: radial immunodiffusion, measures serum proteins, e.g. immunoglobulins. Auchteloni double diffusion, fungal and bacterial antigen detection. Complement fixation test, CFT. Principle: this test checks for the presence of antibodies by assessing the ability of an antigen-antibody complex to fix complement proteins. Uses: diagnoses syphilis, viral infections. Enzyme-linked immunosorbent assay, ELISA. Principle: uses enzyme-labeled antibodies to detect antigen or antibody presence. A color change indicates a positive reaction. Uses: diagnoses HIV, hepatitis B/C, hormone levels, used in vaccine research. Immunofluorescence assay, EFA. Principle: fluorescent dye-labeled antibodies bind to specific antigens and are detected under a fluorescent microscope. Uses: detects malaria, plasmodium, syphilis, treponema pallidum. Neutralization test. Principle: measures the ability of antibodies to neutralize a virus or toxin. Uses: diagnoses rabies, polio, diphtheria. Rapid diagnostic tests, RDTs. Principle: antigen-antibody interaction produces a quick visible reaction, e.g. color change. Uses: COVID-19, malaria, dengue diagnosis. Conclusion: serological tests help diagnose infectious diseases, autoimmune disorders, and blood compatibility. Understanding their principles and applications is crucial for accurate diagnosis and treatment.